Unit 5 in AP Biology will be covering heredity. And so this is where we'll start to get into genetics and piece a lot of things that we've discussed so far about cell structure and function and the cell cycle. Now, this lecture is going to focus on meiosis, so we'll be taking a look at the process of meiosis and how it encourages genetic diversity. So our main objectives for this lecture are to go over the basic overview of the process of meiosis and then how it gives us genetic variation. Then we'll take a look at the two meiotic divisions in detail, meiosis I and meiosis II. And then we'll pull it all together and understand why this process is essential for sexual reproduction and why sexual reproduction is important. So before we can understand meiosis, I think it's good for us to review chromosome structure. We touched on this with the cell cycle, but I want to take this maybe a step further to review some of the important parts of chromosomes. So we know that chromosomes basically have two parts, right? They have two sister chromatids, which are the duplicated DNA structures that are joined to each other. And so we can see over on this part of the screen, we can see each of these chromosomes. There's two chromosomes here. They are the same. They're both considered to be chromosome number 2 in humans because of their same size and genetic composition. But each chromosome has a pair of DNA molecules that are condensed into what we call chromatids. Autosomes are the 22 chromosomes in humans that do not determine gender. So there's 22 autosomes. And you can see those are arranged here on this picture of chromosomes based on their size. So chromosome number 1 in humans is the longest chromosome. And you can tell these are homologous because they have the same size and the same banding of genetic composition. So you notice that the smallest is chromosome number 22, right? You can tell they're very small and they have the same little banding pattern there. Now, sex chromosomes are the distinctive pair of chromosomes that help to determine the gender of a human individual. So over here, we can see this particular image is showing the chromosomes from a human female. So there are two X chromosomes, right? We can see they are homologous, they're the same shape, same banding pattern. If there was a Y chromosome, well, then we would see that there would be a male, because on the Y chromosome it would be one X and one Y, because Y has a gene, the Y chromosome has a special gene called the sex-determining region, the sex-determining gene, which basically is responsible for the production of testosterone, giving us a male human. But we will touch more on that when we look at chromosome inheritance at the end of this unit, and we'll take a look at karyotypes and what you're seeing here, pictures of chromosomes in a bit more detail. Now, what we're seeing, what we need to understand as far as meiosis is there's a key difference in meiosis as far as dividing homologous chromosomes in a particular structure. And we'll see that. When we look at meiosis, we're talking about a special type of nuclear or cell division that occurs in your gametes, in your sex cells. So in humans, this would be how or human males, this would be how your sperm is produced in your testes. In females, this is how your eggs are produced in your ovaries. Now, there's some key differences, and some of them have to do with the arrangement of the chromosomes, and that's why I wanted to review chromosomes. But basically, if we take a look at the overview of meiosis, we're starting out with one diploid cell. What does that mean? It has two copies of each chromosome, right? So, so we can see we have a homologous pair of chromosomes prior to replication here. Now the chromosomes are going to replicate, and that's where now we have each chromosome has two sister chromatids. Okay, so this this replication that occurs during S phase then is going to occur prior to entering meiosis I. Why is it called meiosis I? Because this is the first nuclear or cell division that occurs here. Right? So starting out with the diploid cell, we copy the DNA in the S phase, and now we have two homologous chromosomes with sister chromatids. Really, this is no different than what happens in mitosis so far. So we start out here with a diploid cell, sister chromatids. Now, where it is a little bit different is that we are going to separate the homologous pairs of chromosomes. That's a big difference between mitosis and meiosis. We didn't do that during mitosis. We just separated the chromatids. Here, the first division separates the homologs from each other. Then meiosis II occurs. That's where the sister chromatids separate. Well, what's the big deal about this? It looks similar to mitosis, it's just there's two cell divisions. Well, here's the key. So you have two cell divisions that result in 4 haploid cells. That's key, right? We're going from a diploid cell where you have 2 copies of each of the chromosomes in your cells. But the result is, in your sperm and egg, they are haploid. Notice only one N. They only have 1 copy of each chromosome. So we reduce that chromosome number from 46 in human diploid cells now down to 23. Again, we're going to talk more about that later, but I'm just kind of letting you know that's what's happening here. So each haploid cell now is genetically unique. So no 2 sperm, no 2 eggs are genetically well, they could be genetically the same, but typically they're not because there's so many genes, so many combinations. So this in sexual reproduction, like the big picture here, encourages diversity. And we'll talk more about that as we get into genetics. But now let's talk about this whole genetic variation, how it happens. What's going on to make all of your sperm cells and your egg cells genetically unique? Well, one of the key points during meiosis is something called synapsis, where you produce a bivalent. A bivalent is where the homologous chromosomes. Now, these may appear to be different colors, but we know that they're homologous because they have the same size and same banding patterns, right? So this is basically 2 identical chromosomes. If you recall in mitosis, all of the chromosomes lined up single file in the middle of the cell, and then the sister chromatids were separated. That was mitosis. In meiosis, we form a tetrad or a bivalent. The homologous pairs line up 2 by 2 during metaphase, and that's a key difference. When the chromosomes are lined up during this synapsis, or this kind of connecting part, you basically have a complex formed, and something very significant can happen, because now, because they are more or less conjoined during this bivalent or synapsis event, they can exchange genes. So you can see that here. It's like these 2 homologous chromosomes have crossed their arms or their legs and have basically traded pieces of DNA. And so notice at the end, this original chromosome that was all red now has a blue portion. Again, still, let's say chromosome number 1, but they've exchanged genes. And so significantly, this maternal chromosome, and that's why we have 2 copies of each chromosome. One comes from mom, one comes from dad. And so what's happened here is the maternal chromosome has exchanged some DNA with the paternal chromosome. Basically, that's pretty neat. When you look at your sperm cells and your egg cells, they have a combination of genes from your mom and dad, so that your offspring aren't just going to inherit, well, all of their granddad's traits. They could inherit a combination of them because of this interesting process of crossing over. Let's see what that looks like in this video. This will give you a nice demonstration of how this leads to genetic diversity.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for two genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have four different genetic combinations. Two will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other two will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Let's take a closer look at the detailed events of meiosis I. Again, this will seem very similar to mitosis, but there's some key differences that we're going to look at. So during the first meiotic division, we have a prophase I, just like we had prophase in mitosis. But during meiosis I, we call it prophase I. And the same events are happening. Basically, we see the homologous chromosomes become apparent, and the nuclear membrane is starting to dissolve. We're starting to see the spindle apparatus form, which leads into prometaphase. Now the nuclear envelope is completely dissolved, and we can definitely see the microtubules starting to connect to the kinetochores of the homologous chromosomes. Here is where your key difference is between meiosis and mitosis. During metaphase I of meiosis, notice the homologous pairs of chromosomes line up. And so then, as a result, in anaphase I, the homologous pairs of chromosomes are pulled apart from each other. In mitosis, the chromosomes lined up single file, and it was the sister chromatids that were pulled apart. So this is a key difference here that occurs in your reproductive cells. And then, of course, you have telophase and cytokinesis. All right, so those are the events of meiosis I. Here's a little video that will give you an animation of what is occurring.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the two successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase I of meiosis I begins with the condensation of chromosomes and the vesiculation of the nuclear membrane. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase I, the spindle apparatus continues to form, and bivalents become attached to kinetochore microtubules. One pair of sister chromatids is connected via kinetochore microtubules to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase I. The chromosomes align at the metaphase plate during metaphase I. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase I involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in two cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis I, and after a period of interkinesis, the two daughter cells start the second cell division cycle called meiosis II.
This video will show you how the random orientation of chromosomes will lead to genetic diversity in your reproductive cells.
During metaphase I of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
So you can do that math with human chromosomes. We have 23 pairs. So that video just showed you 3 pairs. What would the possible combinations be if there were 23 pairs? We can share that information and that answer in class. Now let's take a look at the second meiotic division, or known as meiosis II. And so now we just repeat the phases that we learned in our mitosis lecture last unit. So we start out with prophase, but it's occurring during meiosis II. We call that prophase II. And notice now we have those duplicated haploid cells from meiosis I. They are both going to go through the prophase stage. Then they'll enter prometaphase II, where a new spindle apparatus forms, and then metaphase II. Now, notice, because we don't have any homologous chromosomes, now the chromosome pairs, or the sister chromatid pairs, line up along the metaphase plate in a single file fashion. Anaphase II now separates the sister chromatids, and then in telophase II and cytokinesis, we end up with 4 cells that are all haploid. They have half the number of chromosomes that they did in the beginning. Here is a video that will animate that for you.
At prophase II, the chromosomes condense again, and the nuclear membrane vesiculates. During prometaphase II, the spindle apparatus forms, and the sister chromatids become attached to kinetochore microtubules. In this case, a pair of sister chromatids is attached to both poles, not just one pole as in prometaphase I. At metaphase II, the sister chromatids are aligned along the metaphase plate, with each chromatid being attached to one pole. The sister chromatids separate or disjoin and begin moving to the poles of the cells during anaphase II. During telophase II, the nuclear membrane reforms and chromosomes decondense. In animal cells, a cleavage furrow is formed that causes cytokinesis or cellular division. As cytokinesis is completed, meiosis II results in the formation of 4 haploid cells. Thus, meiosis begins with a diploid mother cell that has replicated its chromosomes, and ends with 4 haploid cells containing 1 set of chromosomes each.
And here's a video that will review the overall process of meiosis for you. I know it's replication, but like they say, with replication comes retention.
In prophase I, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase I, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Now in this video, we're going to summarize what makes meiosis unique and different from mitosis.
Although mitosis and meiosis have much in common, meiosis has three unique features: synapsis, homologous recombination, and reduction division. Following chromosome replication, homologous chromosomes, or homologs, pair all along their length. The process of forming these complexes of homologs is called synapsis. The second unique feature of meiosis is that genetic exchange occurs between the homologous chromosomes while they are physically joined. The exchange process that occurs between paired chromosomes is called crossing over. The third unique feature of meiosis is a reduction division. At the end of meiosis, each cell contains only half the original complement of chromosomes. Because of the crossing over that occurs during the first division, the sister chromatids in meiosis II are not always identical. Omitting a chromosome duplication before meiosis II produces haploid gametes.
Let's take a closer look at some of the differences. Here's a side-by-side video that'll show you the differences, and then we'll talk about the details in this table here.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
So let's take a look at some key events of the cell cycle and how they differ between mitosis and meiosis. So the first is DNA replication. This, of course, in the cell cycle occurs during the S phase or synthesis phase of interphase. Well, this occurs prior to mitosis and also prior to meiosis I. Notice it does not occur between meiosis I and meiosis II, between the two meiotic divisions. What about that synapsis, when the homologous chromosomes come together? That does not occur in mitosis. It only occurs in meiosis I. But notice it also doesn't occur in meiosis II. All right. Now, what about crossing over? Well, crossing over could occur during mitosis, but it's extremely rare. It's very common for bivalence, when you have a true synapsis that is occurring, to happen during meiosis I, and it rarely occurs during meiosis II. What kind of separation of chromosomes occurs during anaphase? Well, in mitosis, sister chromatids separate. In meiosis I, homologous chromosomes separate. And in meiosis II, we're back to separating the sister chromatids. And what's the end result of these different types of cell divisions? Well, in mitosis, you have two daughter cells that are diploid and genetically identical. At the end of meiosis II, you have four daughter cells that are haploid. They have half the number of chromosomes that the parent cell had, and they are also genetically unique. This is all key to reproduction. So basically, in sexual reproduction, you have two haploid gametes will unite to fertilize a diploid cell or a zygote. So let's look at what's going on here in this diagram, okay? So here we, of course, have a human couple, right? Here is a female with her ovaries and a male with his testes, right? They are multicellular organisms that are diploid, meaning they have two copies of each of their chromosomes for a total of 46. So there's 23 unique chromosomes in humans. Remember that from the beginning of the lecture. And so they have 23 pairs. And we have pairs of chromosomes because one comes from mom, one comes from dad, right? So this gentleman here has 23 pairs of chromosomes for a total of 46. Half come from his dad and his mom. And this woman here, same thing. She has 46 total chromosomes, 23 pairs, one from mom, the other from dad, her dad. Okay, now, in the ovary and testes, meiosis occurs. In the ovary, meiosis will result in the production of haploid egg cells. Notice they go from being 2N to 1N, which gives us only 23 total chromosomes. Now, in the testes, the male produces haploid sperm cells. And so again, only 23 chromosomes, not arranged in pairs. So notice this is a key feature. We've gone from 46 chromosomes in each of these individuals to now they only have 23 in their reproductive cells. When fertilization occurs, a diploid zygote with a total of 46 chromosomes will form. And then as a result of mitosis, will grow and develop into an adult. So if meiosis didn't occur, let's take a look at what would happen to chromosome numbers. If the chromosome number was not reduced in sperm and egg cells, they would each have 46 chromosomes, and the diploid zygote would end up having 92. So that would be a problem. Too many chromosomes for a human. They are supposed to only have 46. So this is why in sexual reproduction we have a haploid and a diploid stage here. So a lot is going on inside your bodies that you're not quite aware of. And so this is very important for producing genetic variability, because remember, we looked at all the recombination of genes, and we looked at the crossing over and the random assortment of the chromosomes lead to each sperm and egg cell having a unique genetic combination. And that's why offspring will inherit similar traits from their parents, but it's usually a combination of two, and sometimes it's a little bit difficult to predict exactly what traits will be inherited and from whom. So this genetic recombination is the exchange of genetic material as a result of sexual reproduction. And as we go through the course, we're going to learn why this genetic variability is indeed so important to not only species, but also to ecosystems. It's pretty interesting. Let's take a look at this video that will kind of put everything together for us.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, Meiosis I, halves the number of chromosomes, and in the second division, Meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only 1 of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.